Perfect. You're all set.
Okay. Hi, everyone. My name is Nguyen. I'm a bioinformatics PhD student at Case Western Reserve University's School of Medicine. And today I'm going to describe our design pipeline to extract the metatranscriptome from human plus RNA-seq data, as well as highlight the importance of parameters fine-tunings and data processing for result analysis. Um, the human microbiome, often referred to as our second genomes, impact human physiologies both in health and disease. Study has suggested that study has suggested the existence, metabolic activities, and functional importance of the intratumor microbiome. That each subtype of tumor has a distinct microbiome compositions, and that they are different from normal human microbiome. However, these findings are limited to solid tumors. Why acute myeloid leukemia, which is the cancer of blood and bone marrows, are liquid tumors. This cancer produce immature cells that develop into malfunctioning white blood cell and has a 5-year survival rate of 27%. Thus, AML constitute a huge threat to the human health, and its microbiome at the tumor site remain relatively unexplored. Previous work in our lab has mined microbiome sequence from deep shotgun DNA sequencings of the blood and bone marrow of 1870 myeloid malignancy patients and the bone marrow of the 12 healthy donors. Using relative samples genera relative abundance, the results indicate that the microbiome compositions of the normal control are distinct from the myeloid malignancy samples. Um, Intrigued by this, we are curious in whether the circulating microbiome contents carries any functional activities within the tumors, and thus we decide to investigate the RNA contents in AML patients. We thus turn our focus to the BEAT AML projects, which is a large collaborative effort led by Oregon Health and Science Universities, and it offered the largest to date data set on primary AML samples, which include genomics, transcriptomics, clinical and drug response data. Uh, the data I analyze are collected from the blood and bone marrow of 411 AML patients, 21 healthy controls, and 12 technical replicates come from one single healthy bone marrow donors. These samples are divided into 12 groups, which explain why there are 12 technical replicates. These 12 technical replicates will play an important role in our downstream quality controls of the data. Here I describe the pipeline that we designed. The first part of the figures describe the data collections process of the BEAT AML projects, where they purify mRNA from bone marrow samples and subject it to sequencing. After we obtain the RNA sequencing data from the BEAT AML projects, we search it against the Kraken2 database to filter out human consequence and no contaminants. With the remaining microbiome sequence, we process them to address any arise batch effect before preparing them for downstream analysis. Kraken2 is a classification system that uses exact k-mer mesh for high accuracy and fast classification speed. In this extremely simplified examples, I have an input sequence of 40 nucleotides in length. Kraken2 will break them into 6 possible k-mers of um, 35 nucleotides in length and search it against it database. So for in these examples, the first exact k-mer was mapped to species one. The second k-mers was mapped to species two. The third was mapped to species three. Fourth species one, fifth species four, and the last k-mer was not found in the database. Um, the taxonomic level with most k-mer classified in this input sequence is species one with two k-mers, the first and the fourth. The classification score for species one in this input sequence is calculated by dividing the number of k-mers that got mapped to species one, which is two, over all of the possible k-mers, that is six. So 2 over 6 is 0.33. Kraken 2 then decide whether this input sequence is classified or not based on its confidence parameter. Briefly speaking, if the set confidence parameters are lower than the classification score, in this case if it's zero point 0.1 lesser than 0.33, the sequence will be classified as belong to species one, and vice versa. If the set confidence is higher than the classification score, so for example, zero point 0.5 greater than 0.33, the sequence will be unclassified by Krakens. Um, I highlight this confidence parameters because it is important, as the default values for confidence parameters in Kraken tools is zeros. Um, when we offer the default parameters for our classifications process, we coming through the output we receive, we observe obvious false positive classifications. Here is an examples of a false positive observed in my data. This input sequence at the default confidence parameters for Kraken 2 zeros was classified as belong to a feline sarcoma virus, which should not be present in my blood and bone marrow, in the beat AML blood and bone marrow data. Blasting this sequence also suggests that it isn't really belongs to this species, and that it more than likely be a false positive classifications. Um, with the default confidence value of zeros, there are these obvious false classifications account for about 0.01% of our total classifications reads. And since we are dealing with low biomass samples, we decide to not underestimate these false positive. After many trials and errors, we decide to increase the confidence parameters to zero point 0.5, which does remove all of these false positive classifications that got mapped to virus that should not belong in the samples, and increase the quality of our classifications. After after that, we offer zero point 0.5 as our confidence parameters for the classifications. Using the relative abundance of genus read in each samples, that is when we normalize the genera reads based on only microbial compositions. We observe some batch effect that it shows a signs of batch effect when samples within each sequencing run seems to cluster closely together. An additional problems when we normalize the data only by microbial compositions is that our 12 technical replicates doesn't seems to cluster together. We reasons that because these 12 technical replicates belong to one person's, they should be grouped closely together, and that thus our data warrants further data processing step. Um, we then decide to account for the read depths in our sample. Within each samples, we recalibrate the genus read counts such that the normalized genus read counts now is calculated by dividing the read classifier to the genus level to the total reads mapped to human in the samples times 1 billions. And then after we reperform principal components analysis, um, we observe that this our normalizations approach seems to reduce the observed batch effect in sequencing run, and it also bring the 12 technical replicates together. From the normalized genera read counts, we also perform uh, breakers, calculate breakers dissimilarities, and visualize the output using principal coordinate analysis. To our surprise, the results seems to indicate that principal components analysis show better grouping of the 12 technical replicates than the principal coordinate analysis. We are intrigued by these findings, uh, and why we are not satisfied that the 12 technical replicates doesn't seem to group together in the principal coordinate analysis space. We were able to show that we found that the breakers distance in healthy control case are much more similar than comparing the their distance to the acute myeloid leukemia patients. Um, as mentioned, why we are not satisfied that the principal coordinate analysis doesn't show grouping of the 12 technical replicates. We reasons that the data is relatively ready for the next step of downstream analysis, while simultaneously keeping tab of potentially more suitable approach to transform the data. Um, we believe that the data is relatively ready for the next step, which include investigating relationship between blood microbiome of patients and survival status, because we have the clinical data of these patients. We because we are also interested in the functional activities of microbiome contents. We would also want to map the data we have to a database of bacteria's genes to see what genes got and pathway that is highly expressed in certain subgroup of AML patients. And finally, we would like to be able to validate the findings with survival status and blood microbiome compositions and functional microbiome compositions within a different cohort. And with that, I conclude my presentations and I'm happy to take questions.